Is it possible to transmit DNA information without the DNA itself, just through electromagnetic waves? This radical idea lies at the heart of the controversial Montagnier hypothesis, named after Nobel laureate Luc Montagnier. In a series of experiments, Montagnier claimed that highly diluted solutions of bacterial DNA emitted low-frequency electromagnetic signals that could imprint information into pure water nearby. Even more provocatively, he reported that this signalized water could later be used to reconstruct the original DNA sequence through PCR or polymerase chain reaction, as if the DNA had been teleported. But how can this be? DNA is a physical molecule, so how could its complex structure be recreated without any physical template? Montagnier suggested that water might hold a kind of memory, capable of storing and transmitting biological information through coherent electromagnetic fields. Ideas echoing back to controversial claims about homeopathy and quantum coherence in biological systems. Most mainstream scientists remain highly skeptical. Critics argue that these results lack reproducibility, may suffer from contamination, and contradict well-established molecular biology. Yet the hypothesis sparks deep questions. Could there be undiscovered mechanisms of biological communication involving waves, not molecules? Are quantum or electromagnetic effects playing hidden roles in life processes? While there is no widely accepted evidence for DNA teleportation, the very idea pushes the boundaries of how we think about information in biology, reminding us that nature might still have surprises waiting at the edge of the known.
Can a strand of DNA send its genetic code across space without a single molecule moving? Can water somehow remember DNA and rebuild it from nothing but light and expectation? These questions sound like science fiction or perhaps even pseudoscience, but they come from real experiments conducted by a Nobel Prize-winning scientist, Luc Montagnier, who stirred controversy with a bold and deeply puzzling claim that DNA can transmit its information through electromagnetic waves, and that this information can be received and reconstructed elsewhere. Let's start with what was claimed. In Montagnier's lab, a fragment of DNA was diluted repeatedly in water, so much so that, by all accounts, there should have been no DNA molecules left in the sample. Then something strange happened. The water began to emit a low-frequency electromagnetic signal, one that appeared to carry some sort of biological signal. Montagnier's team recorded this signal, sent it electronically to another lab, and played it near a vial of pure water. That water, when subjected to a standard PCR reaction, a technique used to amplify DNA, somehow produced a DNA sequence almost identical to the original. How could this be? If there were no DNA molecules present, where did the information come from? Montagnier suggested that water can form complex structures, nanostructures, that act as a kind of ghost imprint of the original DNA. These structures, he believed, could store genetic information and transfer it through electromagnetic signals. In his view, DNA wasn't needed physically if its electromagnetic blueprint was preserved and transferred correctly. To visualize this, imagine removing a sculpture from a mold, yet still using that empty mold to make a perfect copy somewhere else, except the mold isn't made of clay or metal but water, and instead of being shipped, it's recreated by a pattern of sound or radio waves. This sounds extraordinary, but is it true? Most scientists would say no. Despite the dramatic nature of these claims, they have not been independently reproduced under controlled conditions. Critics argue that contamination, microscopic amounts of DNA accidentally present in the lab could explain the results. Others point out that no known physical mechanism could account for information being stored or transmitted in this way by water alone. And yet, the idea continues to spark curiosity. Could water, under certain conditions, retain some structural memory of what it once contained? Is there a hidden layer of information in biology that we haven't yet uncovered? And could electromagnetic fields play a larger role in molecular biology than we currently understand? There are historical precedents for controversial ideas that were later validated, but also many that were disproven. In this case, while the scientific consensus remains skeptical, the topic continues to fuel debates, alternative theories, and even speculative technologies. Some liken this to the idea of quantum entanglement or teleportation in physics, where particles affect each other instantly over distance. But even in quantum mechanics, information is not transferred without a medium or mechanism. With DNA teleportation, we simply do not yet have a solid, testable framework to explain how genetic information could travel without physical molecules involved. So, can DNA really be teleported? Based on current scientific understanding, the answer is no. But the fact that this question can even be asked based on real experiments, however disputed, shows just how much we still don't know about the invisible forces that might shape biology. And maybe that is the real value of Montagnier's work. Not that it proved DNA teleportation, but that it challenges us to keep asking what's possible in a universe that often turns out to be far stranger than we expected. The DNA Teleportation Paradox arises from controversial experiments conducted by Nobel laureate Luc Montagnier, suggesting that DNA information can be transmitted through electromagnetic signals without the physical transfer of molecules. According to his reports, DNA sequences from one tube appeared to reconstruct themselves in a distant container of pure water after exposure to low-frequency electromagnetic fields. This phenomenon, if validated, would imply that genetic information may exist in a form detached from its molecular substrate, defying conventional models of inheritance, replication, and biochemistry. Montagnier's team claimed that DNA diluted in water and exposed to electromagnetic waves could induce the formation of identical DNA sequences in another test tube of pure water, merely through signal exposure and subsequent PCR amplification. It's like burning a book in one room and somehow the words reassembling themselves on blank pages in another room, transmitted not through ash or smoke, but through invisible waves. Montagnier's hypothesis rests on the idea that DNA emits specific electromagnetic signatures when diluted and agitated in water, and that these signals can imprint structural information onto water molecules. When these imprinted water samples are subjected to PCR, polymerase chain reaction, in the presence of base pairs and enzymes, they appear to reconstruct the original DNA sequence, as if the sequence had been teleported through an informational field rather than physical transfer. A vial of ultra-pure water placed near a container of diluted DNA under electromagnetic excitation reportedly produced DNA fragments identical to the original template after standard amplification protocols. It's like playing a song on a speaker in one room and the vibrations arranging dust particles into the exact shape of the music notes on a sheet in the next room. Montagnier and collaborators invoked quantum coherence, structured water domains, and electromagnetic resonance as possible mechanisms. These ideas intersect with controversial theories like water memory, which suggest that water can hold information about substances that were once dissolved in it. Even after extreme dilution, some propose that DNA acts as an oscillator, emitting low-frequency waves that influence the spatial arrangement of water molecules, possibly forming nanostructured water frameworks that guide the assembly of base pairs. The theory builds on the earlier work of Emilio del Giudice and Giuliano Preparata, who proposed that coherent domains in water can store and transmit quantum information. It's like striking a tuning fork that causes nearby water droplets to self-organize into a pattern resonating with the sound, even after the fork is removed. If proven true, DNA teleportation would radically shift our understanding of biological complexity, suggesting that information transmission in biology is not strictly chemical, but may involve field-based signaling systems. This would imply that biological information can exist independently of its material medium, opening parallels with fields like morphic resonance, biofield theory, and non-local communication. Such ideas challenge reductionist views and point toward a new informational paradigm in living systems. Some alternative researchers link this phenomenon to embryonic morphogenesis, or cellular communication mechanisms, not fully explained by genetics or biochemistry alone. It's like discovering that conversations between cells are not just whispered through chemical signals, but also broadcast on frequencies like hidden radios embedded in biology. Montagnier's experiments highlight the importance of electromagnetic fields as potential carriers of biological information. The energy supplied by these fields may enable structural reorganization of water molecules or activate latent informational patterns. This challenges the traditional view that information must be bound to molecules and suggests that fields may act as scaffolds for organizing molecular assembly, blurring the line between energy, matter, and information. DNA was reported to reappear only when both electromagnetic excitation and PCR reagents were present, indicating that both informational input and energy input were necessary. It's like sending a blueprint by invisible light beams, but needing a construction crew and raw materials for the building to materialize. In information theoretic terms, the claim implies that DNA may be represented as an electromagnetic code capable of being transmitted and reconstituted across space, much like data sent through Wi-Fi. The concept invokes non-local encoding of biological instructions, akin to holography or quantum entanglement, where the whole is contained in each part, and information may not require direct physical pathways to exert influence. The suggestion that phantom DNA patterns can linger in the electromagnetic field and guide molecular assembly recalls quantum biology discussions around wave function encoding and informational fields. It's like copying a software program over a wireless signal. The hardware is blank, but once the signal arrives, the code reinstantiates itself exactly. If DNA teleportation proves valid, it would revolutionize genetics, regenerative medicine, and bioengineering. It could enable non-invasive gene therapies, DNA information transfer without vectors, or remote control of gene expression. It might also offer new explanations for phenomena like remote healing, biofield detection, or spontaneous regeneration. At the fringe, it touches on ancient ideas of vibrational medicine and informational templates for life. Some speculative researchers envision wave-based medicine, where DNA could be corrected or repaired by applying the proper frequency, restoring genetic function without cutting or inserting genes. It's like fixing a corrupted document on your computer by beaming in the right software patch without plugging in a USB or touching the keyboard. Montagnier's claims have been met with widespread skepticism, primarily due to lack of reproducibility, unverified methodologies, and departure from known molecular biology principles. Attempts to replicate his results independently have largely failed or produced inconclusive outcomes. The scientific community demands rigorous controls, transparency, and falsifiability, and as of now, the evidence for DNA teleportation remains controversial and unaccepted by mainstream science. Journals and reviewers have called for blind, double-controlled studies with independent labs to confirm whether any signal DNA formation is replicable or an artifact. It's like hearing about a teleportation device that works only when one person is in the room and no one else can get it to work. Not enough to rewrite physics just yet. Even if Montagnier's specific results are disproven, the questions he raised remain valuable. Future studies may explore how electromagnetic fields influence molecular self-assembly, whether biological information can be stored non-molecularly, and how quantum coherence in biological water plays a role in health and communication. This field sits at the intersection of quantum biology, biofield science, and systems medicine, offering new frontiers for theory and experimentation. Emerging fields like quantum biosignaling and water-based nanostructures continue to explore how living systems might encode and retrieve information from field effects beyond mere molecules. It's like suspecting that libraries do not just hold books, but that the rooms themselves whisper stories if we learn how to listen in the right frequency.